really is some preliminary research that we discussed earlier today from a study called PEACE. This is a national autopsy study or a post-mortem program that we have established in the United Kingdom, where we are recruiting patients with metastatic cancer to be part of the study. And what it involves is after death and soon after death, we collect tissue from all the sites where the cancer has spread in the body. And there are many different types of cancer research that we can do with these samples, but in particular I'm interested in trying to find out the tumour landscape or the genetics of the cancer, and to define that lethal clone or population of cancer cells that seeds and spreads and results in metastatic disease. So that explains the title a little bit.
When it comes to tracking those back to any points of origin, is that the eventual goal?
So that's where the real power of a study like this would be, being able to compare the very beginning of a cancer towards the end, at the point at which you have widespread secondary cancer. And we're fortunate enough to be able to do this because we have another study called Lung Tracer X. And in the Lung Tracer X study, we collect the primary, the original tumour, the lung cancer, and we follow these patients up very closely for many years. And when their cancer returns and they develop metastatic disease, we go back and we collect samples, and then we're in a position to be able to find out the evolution of this lung cancer from the beginning, primary to metastatic disease.
And are there any hallmarks of change in the genetic landscape or the microbiome which you've identified so far?
So it's still early days. Today we talked about the first 4 patients from the Tracer X study who have gone into the PEACE study with lung cancer. Although PEACE itself has recruited over 100 patients from many different tumour types, but we're already beginning to see some very interesting, fascinating patterns in the way cancer appears to be evolving on a genetic level. So we can see that there are different patterns in which cancer can spread. It can be monoclonal, so a single dominant population of cancer cells that might seed or spawn the metastases. It can be polyclonal, so multiple populations of cancer cells that do the same thing. We don't quite know what that means in terms of clinical outcomes or what it might mean in terms of response to treatment. But the more patients we study, the more we might be able to understand that better. But we're also seeing that some genetic abnormalities might occur early and some might occur later on in tumour evolution. And I'm certain that the more cases we analyse, the more we might understand about those specific genetic drivers or events that dictate metastatic disease and disease progression.
And then is this also being weighed against any treatment history they've got to see if there are any drivers in that?
I think that's a real important question, and certainly something that we're looking at. So every patient who enters the study has a very detailed retrospective medical and drug history taken, in particular the treatments they've received for their cancer. And one of the things we really want to understand is why the last therapy they were on didn't work or wasn't adequate enough to control the disease and at least slow down disease progression. Did they develop markers of resistance? Could it be the genetics? Could it be the microenvironment of the tumour? Could it be the immune system, and so on? And we can answer that in the context of simple chemotherapy. I say simple in that as opposed to targeted therapies. But what's becoming interesting now, especially in the context of lung cancer, is immunotherapy, trying to understand why some patients develop resistance to immunotherapies. And I'm hoping that PEACE will get us that step closer to understanding more about that process.
I suppose finally, in terms of the application and implementation of the idea of, like you say, tracking this information back and observing people in Tracer X, the use of liquid biopsy has been increasing over year by year, and we've heard lots of people talking about the vast array of data that can be stored in biobanking. Are these technologies being brought into the PEACE study as well?
So PEACE is almost like a fancy biobank, although it's much more than that. It's a biobank in the sense that we are collecting tissue retrospectively. We can access archive samples, so a patient may have had tumour resected as part of surgery for their treatment, and that may have been banked in a biobank, and then after death we collect multiple sites of diseased tissue from them, and we can try and compare the two. So it certainly leverages the idea of a biobank, banking samples, but The way in which it's unique and different is that PEACE isn't just banking, we're analysing the samples as we go along. And you mentioned liquid biopsies, that's a strong theme in the study, and it links in well with lung TracerX. TracerX also, there are studies in kidney cancer and skin cancer, similar setup. But in particular in lung cancer, we are longitudinally collecting liquid biopsies to the point of death to try and see how we can track the evolution of tumours and cancer in the liquid biopsies, but in particular in PEACE, at the point where there's widespread metastatic disease, we're trying to look back into the liquid biopsies to see whether there was something in the blood that could have forecast that progression. And what we find at autopsy or at post-mortem, could we find that information also in the liquid biopsies prior to death? And we already know that that's possible. We've seen that in at least one case in the lung TracerX study. And the more patients we look at, the more powerful that data will become.